The first is that when you affect electron transfer, electrons can move in DNA. If you look at a model of DNA, the bases have the pi electrons on the bases are present in a sheet. In fact, people who've done research on electron movement in DNA refer to this continuous sheet of electrons in DNA as a pi way, because they are the pi electrons that are delocalized in electronic configurations around atoms. And you can easily move an electron. You can calculate what an ELF field, as weak as it is, how far it can move. It can move an electron sufficiently to transport it along a hydrogen bond and certainly displace it. The second point about local charging: when you charge up when you move an electron, you'll carry a negative charge to a place that didn't have it before, and you leave behind an uncompensated positive charge. So you're generating charge. And one of the things that happens when you generate charge in a macromolecule is you get disaggregation. And we can conceive of disaggregation in a particular local region of the DNA. And this initiates protein synthesis. Now, I've gone through this rather quickly. I have a longer treatment of the background on this in the Electromagnetic Biology and Medicine, which is a journal edited by Abe Liboff, who was active in electromagnetic research for many years and now edits this journal. But it's a long paper in which I go through the details in case anyone's interested in following it up. I have here a picture from a paper by Aaron Klug, who was a Nobel laureate. This is the Nobel address, in which he shows how charging affects the disaggregation of a protein aggregate in the case of tobacco mosaic virus. Now, the interesting thing is, I don't have a pointer, but if you look at the lower left corner, you see this big aggregate of all these proteins are together. That's at the isoelectric point of the... great, thank you. That's at the isoelectric point of the protein. But as you charge up the protein, it starts to split up, eventually splitting up into individual monomers. Now, what happens is, this is ionic strength. As you decrease the effect of the charge, you can get the thing to reaggregate again by swamping it with the with extra ionic atmosphere. But the point is, this is a general kind of a phenomenon. Aaron Klug studied it in tobacco mosaic virus. We showed this. We did a similar effect in studying the disaggregation of hemoglobin, which is a tetramer, into two dimers. And you can see that as soon as you titrate, you add some more proton, you get the hemoglobin coming apart. Now, a single proton can get these two big hemoglobins, hemoglobin dimers, to separate. My God, that's a lot of energy, because the energy is not in the added proton. The energy is locked up in the structure of the hemoglobin itself and the hydration shells that exist around the hemoglobin. And you utilize the energy that's in that to get this molecule to disaggregate. So this is a long way of saying that this is a possibility, that nature has built in this structure into the DNA, and if you perturb it by changing the charge arrangement on it, it's a relatively easy thing to get it to come apart. And I think this may be what happens. Now let's look at where the DNA actually comes apart. This is a picture of the promoter in a particular heat shock protein, HSP70. And it turns out that there are regions, and these are symbols that stand for the DNA along this chain. This is the actual gene, the coding for the gene, but these are the DNA components in the promoter. These red areas are Ctct bases, and the reason we designate these as special is because we've been able to show that they are indeed special. First of all, let me make the point that you've got two different regions in the promoter. You've got a region that responds to heat, and you've got a region that responds to EMF. The heat response will only respond to heat. If you remove this, it won't be turned on by heat, and if you remove this, it won't be turned on by EMF. So that these are very specific domains, and this is published. And let me mention now about the studies with Ctct bases. these are interesting combinations. They're not that rare. You would expect this particular combination once every 270 times, just by the fact that there are 4 different bases that are used within the DNA. But the C and T have low electron affinities, which means that the electrons can be moved easily, so that they have the right physical properties to do the kinds of things that I've been waving my hands about. Now, there was a recent study published in Science which showed that there is a difference in the fluorescence decay rate between the CTs and the GAGAs, which are on the complementary chains. Now, that means that you got your CTs able to dissipate the energy that they've been fed by these fluorescent stimulation, and these GAGAs hanging on to it. So you've got a system which is unstable, and one will more easily come apart, and while the other one is holding the matrix together. And the other interesting thing about this is that because you've got pyrimidines on one half and purines on the other, they come apart smoothly. In other words, they make a smaller surface. And if you try and calculate the energetics of what that means, it turns out to require less energy because of that particular geometry. So anyway, it looks like that's a particularly good combination. But now let's look at what happens if we take a chunk of this promoter, and we actually took a chunk of the promoter, not from the HSP70, but from this CMYC, which is also turned on by the EMF. And if you take, it was close to 100 base pairs, we took a piece of that out and attached it to a reporter gene. We had 2 different reporter genes, and we were able to stimulate it with an EMF signal, a 60 hertz signal. So we could turn on the chloramphenicol acetyltransferase activity by stimulating that piece of the promoter that we had transferred in there as a kind of EMF sensor. And we did the same thing with luciferase. So we were able to take that particular piece of DNA and show that that was responding to the EMF signal by transferring it to something else and showing that it was able to do that stimulation. No. Okay, I'll try and finish up. I'd actually hoped to add to what I had. As the conference started, I saw there was more material that one wanted to do, but I guess I'll have to bring that into the discussion. But anyway, let me continue with the 2 minutes minus what I've said now. I'll bring in leukemia epidemiology, which we have and the tie-in with DNA, that we know that there's been a lot of leukemia epidemiology that has linked the possibility of EMF with the possibility of cancer. There was a recent paper published in China, from China, in leukemia and lymphoma, which shows that there are mutations in DNA repair genes that increase the risk for leukemia. And the level at which they say the risk increases, the odds ratio increases to over 4 at a field of the order of less than 2 milligauss. So if I can summarize this all, we have that DNA damage is believed to lead to cancer. That's pre-EMF studies. That exposure to EMF increases the risk of cancer. We've seen that from epidemiology studies. That EMF stimulates DNA protein synthesis and DNA damage from strand breaks. And we have shown that there are specific sequences that interact with EMF, and that mutations in repair genes increase the risk of leukemia at 1.8 milligauss. So there is a plausible scientific mechanism that one can say. And if you talk about the absence of a mechanism, maybe they're talking about RF, but even at ELF, one can put together a series of scientific findings that hang together as certainly as a plausible proposal. And I'd like to end with the fact that a group of us put together the Bioinitiative Report, which pointed to the need for a biologically based precautionary principle, or precautionary exposure standards. And one of the criticisms against this report was that it was put together outside the bounds of normal science. Well, we've gotten the same group together, virtually intact, and we've added a few, and we put out a new bioinitiative, in effect, in an issue of a journal, complete with peer review, and we now have an updated report in a peer-reviewed journal with additional scientific evidence that continues and makes the case again for a precautionary exposure standard. So I'll end there. Thank you.